In this video, we are going to explore NMR spectroscopy, or nuclear magnetic resonance spectroscopy. So in order to understand what NMR spectroscopy is, we first need to understand that some nuclei of atoms, so some examples are protons, C13, F19, and 31 phosphorus, have this property that's called spin. So we're not going to get into too much of the technical details about what this property of spin is, but you can think of spin as the nucleus is spinning, and when you have something with a charge that starts spinning, it actually can act like a little tiny bar magnet. And so when you put that bar magnet into an external magnetic field, the nuclei spin is either going to align itself with this external magnetic field, or it's going to align itself opposed to this external magnetic field. So when it aligns itself with the magnetic field, as in this first image here, then that's a pretty low energy state because they're not opposing one another. Whereas when it aligns itself opposed to the external magnetic field, it's at a much higher energy. So in order to be able to interpret an NMR spectrum, it helps to know a little bit about how an NMR spectrometer works. So the image on the side here is an example of an NMR spectrometer. They are quite large. There are a few different sizes based on the size of the magnet, but you can actually sort of see the ladder in the back here. So that just kind of gives you an idea of the magnitude of the size of one of these machines. And so when you are doing NMR spectroscopy, your sample is placed in an electromagnet. So we've got the magnet here, and then we've got some coils here, and the electricity is then generating the magnetic field. And so once your sample is placed in here, similar to other spectroscopy techniques, the field strength here is going to be varied through the radio wave part of the electromagnetic spectrum. And when the radio waves hit the exact frequency needed to make the nuclei flip, so that their spin goes in the opposite direction, so it transitions from that low to high energy state, something called resonance happens. And when resonance happens, that is detected on the machine and it's recorded in a spectrum. So we can then get information about the environments of these different types of nuclei within the molecules, and specifically, we're just going to focus on protons and the information we get about the environment of the protons within that molecule. The position of the NMR signal relative to a standard, and we'll talk about the standard in a little bit, but it is recorded as something called a chemical shift. So it's given as this lowercase Greek delta letter, and it's measured in parts per million. And so when the nuclei flip and you get that resonance, the signal that is recorded is the chemical shift. And we're going to look at just the chemical shifts of protons. And so protons can be in different chemical environments, so they are going to produce different signals in the spectrum depending on what chemical environment they are in. All of this is measured against a standard, and one of the typical standards that's used in NMR spectroscopy is this tetramethylsilane, or TMS. And so everything is measured against this molecule here, and it's given a chemical shift value of 0. Its chemical shift is not actually 0, but we normalize all of our data to this standard. There's lots of reasons why. We're not going to go into the details in this video here, but it's a really great standard for us to measure our data against. So here's an example of an NMR spectrum of ethanol. And so what you end up seeing for ethanol is 2 peaks on the NMR spectrum. You get one at a chemical shift just above 2, and you get another one around 10. And so these 2 different peaks are based on the fact that all of the protons within this molecule are in one of 2 different chemical environments. And so when we talk about chemical environments, we talk about where the protons are in the molecule and what else they are connected to. So the one proton connected to the aldehyde group, you would say that's in one chemical environment. Whereas the other 3 protons are all connected to a carbon that's part of a methyl group, which is connected to the aldehyde. So these 3 are all in the same chemical environment, which is why we see 2 peaks in this spectra. We can also get some more information from something called an integration trace. So when an NMR spectrum is recorded, the NMR spectrometer, or the computer connected to it, can also calculate the area under each peak to produce something called an integration trace. So this is additional information you would get with your NMR spectra. And what's really neat about this is that the area under each peak is going to be proportional to the number of protons that are in that environment. So what you can do with this integration trace is you can literally go take your ruler and measure the distance from the bottom to the top. So we would measure this distance here, and then we could go and measure this distance to this distance here. And when you make that comparison, you can see that it's in a 1:3 ratio. So what that means is that for every 1 proton in this peak, you're going to have 3 protons in this peak. So it does give you a little bit more information about the structure. And that corresponds nicely with the structure of ethanol given here, right? Because you have 1 proton that's in the aldehyde group, and then you have the other 3 protons which are part of the methyl group. We're going to take a look at a couple of examples now. The first one here is that we want to suggest the number of peaks and the ratio between the areas under the peaks in the NMR spectrum of the following 2 molecules. So what you want to do first is you want to draw out your molecules. And I highly recommend that you do a full structural diagram because you want to see all the connections of the hydrogen atoms. So let's start with 1,2-dichloropropane. And we'll draw this out. So propane is 3 carbons. We've got a chlorine on 1. We got a chlorine on 2. And then the rest are hydrogens. So we're just going to fill that in here. Okay. And then what we want to do is we want to look at all of the protons in this molecule and consider each of those for our environments. If we started the carbon on the end here with the 3 protons, we would say that these 3 protons are all in the same environment because they are all connected to the carbon, which is connected to the next carbon that has the same thing. Okay, so those 3 would all be under 1 peak. The next carbon has a hydrogen attached to it, so this would be a different peak. And there would only be 1 proton in this peak because this hydrogen is attached to a carbon, which is then attached to a bunch of different things here. And then the last environment we would have, or the last peak we would have, are these two protons here. And they are in a different chemical environment again, because they're connected to a carbon that's connected to a chlorine and the rest of the molecule. These protons are different than this proton here on the second carbon, and it's because of the connections that are adjacent to that molecule. So that being said then, we would have 3 different peaks, and it would be in a 2:1:3 ratio. All right, so our second example, butane 2-ol. We've got 4 carbons, and then we've got an OH attached to carbon 2, and the rest are protons. So we're just going to fill all those in here. Okay. And then we're going to go through with the same considerations. So taking a look at the 2 ends carbons, we've got all of our protons here. And you might think that those 3 protons there on that end carbon and the other 3 protons on the other end carbon would be the same chemical environment, but they're actually not, because if you take a look at what the carbon is attached to with those protons, it's attached to different things on either side. So those 3 from the first carbon and the other 3 from the last carbon would actually be in slightly different peaks because they are in slightly different chemical environments. Then you've got the next carbon here. So those 2 protons would be in the same environment, so they would give a peak. You've got the next proton here on this carbon. That would give another peak. And then finally, you got this last proton here on the OH, which is in its own chemical environment too. So you've got 5 peaks for butane 2-ol, and it would be in a 3:1:1:2:3 ratio. So let's just write that down. To 1:2:3 ratio. It doesn't really matter how you write your ratios here. What matters is that you identify where those different chemical environments are. Our second example is a little bit different. So we've got the NMR spectrum of propan 2-ol shown here. Part A wants us to draw the full structural formula of propan 2-ol. B, explain why there are 3 peaks. And C, explain what information can be obtained from the integration trace. So let's start with drawing the full structural formula for propan 2-ol. So we've got 3 carbons. We've got an OH on carbon 2. We've got an H. And then we've got 3 hydrogens there and 3 hydrogens here. So now it wants to know why there are 3 peaks. So if we kind of go through the same type of analysis again, you've got 1 peak that's going to be coming from the middle carbon with the proton attached there. You're going to get a second peak for this hydrogen here on the OH. And then if we take a look at the CH3 groups on both ends, they would all end up being in the same chemical environment. And that's because that carbon is attached to the same thing on either side. Basically, the structure is symmetrical, so all of those protons would be in the same kind of chemical environment. So you're going to get 3 peaks. You're going to get a peak for the blue ones, you're going to get a peak for the yellow proton, and a peak for the green proton. Finally, information that can be obtained from the integration trace. We could go through with our ruler and measure here. So we could measure this distance, we can measure this distance, and then finally we can measure this distance here. So if you were to do that, then what you would notice is that you get a 1:1:6 ratio, which corresponds to the fact that we have the yellow proton, which is 1 in one chemical environment, the green one, 1, which is in another chemical environment, and then the other 6 protons, which are in its own chemical environment. So you can actually match up those blue ones to this peak here because it's a 6 ratio. The other two, you would need a little bit more information to match those up, and that would be coming in a different video.